make sure you tell congratulations to Dr. Indy Gill, who was just announced top 100 in Time magazine for his work on bladder transplantation. Just announced. Congratulations. Yeah. So now it's our pleasure to introduce Dr. Atala, who's legendary in his research on tissue healing processes, and he's going to talk about body on a chip technologies for drug development, toxicity testing, and cancer treatment. He is the force at Wake Forest in North Carolina.
Thank you so much, Dr. Thomas. Appreciate it. Great to see you, Dr. Persky, Dr. Hamilton. It's good to see all of you. And let's see, it's not coming up on the screen. Hi. It's good seeing everyone today, and just wanted to give you an overview of body and chip technologies for drug development and personalized medicine. So as you know, when we talk about Regen Med, as we talked earlier this week, we can use either cells or scaffolds or enabling technologies to create these tissues and organs. And the strategy has been useful to create tissues and organs that we have implanted into patients over the last couple of decades. In 2002, by the time that we put in a number of tissues and organs, we released a white paper that stated, why don't we use these technologies and reduce them for personalized medicine and drug testing? And I'm going to say it got zero reception. No one, people thought it was science fiction. In 2010, we were giving a talk in Washington, D.C., unbeknownst to us, the head of innovation for the FDA was there, and he approached and said, well, you guys are putting these tissues and organs into patients. Have you ever thought about miniaturizing it for drug development? I said, yeah, we put this white paper out in 2002. So he said, can I come visit you? I said, sure. So he spent a day with us in North Carolina, and at the end of the day, he said, we got to fund this. But, we at the FDA, we're not a funding agency, so who do you think will fund it? And, I talked to him about, well, it's not going to be the NIH. It's too risky for them. So maybe, things like DARPA or DITRA. And so why was the FDA interested in this? Because the hard reality is that companies spend millions and millions of dollars developing these technologies. By the time they get to a patient, 90% of technologies that reach phase 1 clinical trials never make it out. 90% that enter a phase 1 clinical trial in terms of drugs. Why is that? Mainly because 2D cultures are not representative of normal human cells, and certainly not cell lines. And animals are also not great models. How many times have we cured cancer in a mouse? So we were very lucky. The government released 3 funding opportunities fairly quickly. One through DARPA for $8 million, several through the NIH, and then this large one for $20 million, which we were lucky to get. And through this grant, we basically used our 3D printing strategies to help standardize these technologies. So how do we create these, what we now call organ tissue equivalents, or organoids, body in a chip technologies, which are, remember, we're just attacking it from a regenerative medicine strategy. So this goes back to over 15 years now. And so the strategy is basically you take cells, normal human cells, just like you would to create a tissue for a patient, all the cells that are present in the human body in the same proportion, all human, all primary. And then we use the extracellular matrix, just like we would to create a tissue. We take a discard organ like the liver. We use very mild detergents to wash cells away. We then lyophilize it, digest it, create it into a bioink, and then, because even the stiffness affects the properties of this. And what's the outcome when you use this strategy? Because then you can have really high fidelity. You can even create these rudimentary bile canaliculi. Now the functionality, long-term, you can see secretion of albumin, urea, et cetera, for over 30 days, which is pretty strong. And this is just when we started doing it, right when we started doing these. So organoids are not created equal. Like, you can call a bunch of hepatocytes an organoid, but the liver is much more than just hepatocytes. That's why all those cells are there. And the ECM also plays a role. And that's why this was important. We can do the same thing for 3D miniature hearts. These are miniature hearts, beat about 60 beats per minute. Once they start, they last for over 6 months. We can give them drugs to either slow down these hearts or speed them up. Basically, in a similar dose response as humans, we can create miniature heart attacks by depleting oxygen. We can actually create lung models using layers of epithelial cells and endothelial cells, but we can also use the immune cells with macrophages, neutrophils, etc. You can see here, it's a good slide to show you the difference. On the top left, you see what a 2D culture looks like. On the bottom left, you look what a native lung looks like. In the center, you see what these 3D organoids look like. And you look at epithelial height, cilia coverage, mucus production, basal stem cells, it replicates much more than native tissue as it would 2D culture. So here's when we put 3 of these together. So in one system, we integrate lung, liver, and heart. Now, we all know that bleomycin is lung toxic, right? We use it for chemotherapy all the time. We know it has toxicity to the lung. So we placed the bleomycin as a testing system. This is back in 2012, so we've been doing this for a while. We basically exposed this to a drug, bleomycin, and what we found was that unexpectedly, the heart stopped beating. And we couldn't understand why the heart stopped. I mean, the lung did show toxicity, but so did the heart. And we thought we made a bad batch of heart, so we put a new heart batch in, and guess what? They still stopped beating. We took the liver out to see maybe it was a metabolite from the liver. It still stopped beating. So we couldn't figure out why bleomycin was showing cardiac toxicity, which had not been shown in humans before. So we basically looked at this, and we started to look at lung inflammation. Turns out when the lung starts to show inflammation, it starts to secrete interleukin-1 beta, and that is toxic to the heart. And so now we know that, in fact, bleomycin is also toxic to the heart at at high levels. The reason we're not seeing it clinically is because you're pulling a drug before you start to see this toxicity. All right, so here's the brain models that we created, and this shows the very first time that what could show an intact blood-brain barrier entirely through human cells without using transwell membranes or silicone membranes or anything of the sort. And these, how do we make them? All 6 major cell types from humans, primary cells from the brain. And you can see here we can create miniature strokes by depleting oxygen. And here's another. In this particular chip, we integrated 6 organ tissue equivalents. We had lung, liver, heart, blood vessels, brain, and testes. And in this particular system, we used the drug ifosfamide. And the drug ifosfamide is another alkylating agent, chemotherapy agent, which we all know is neurotoxic. And so the reason why it's neurotoxic is it gets metabolized by the liver to secrete ifosfamide. To secrete chlorosaldehyde. So what we do here is, you can see here that if we don't put any drug, all the organs are doing fine. If we then, and remember this is an integrated system, so if we place the drug onto it, it's sure enough, we show toxicity to the brain. But if we take the liver out, doesn't get metabolized, you don't show the toxicity. All right, so how do we do with the program? Well, it's a $20 million program. So did Detra get their money back? Well, guess what? This is a drug called Hismanol. It was in the market for 11 years as an over-the-counter drug for allergy medications, as an anti-allergy. It was tested in 2 cell lines, no toxicity. 2 animal models, no toxicity. Phase 1, 2, and 3 human clinical trials, no toxicity. Gets released to the market 11 years later. It's an over-the-counter drug. They finally connect the dots, heart block. So we took the drug, tested our system. Within 2 weeks, we knew the drug was toxic to the heart, same mechanism of action, similar drug dose response. Here's a similar finding, but more tragic. This drug is called Resolin, anti-diabetic and anti-inflammatory. It had been tested. You could see here that this drug had been tested in 2 cell lines, 2 animal models, phase 1, 2, and 3 clinical trials, no toxicity, gets released to the marketplace within 3 years, 63 deaths due to liver failure. We took the drug, tested in our system. Within 2 weeks, we knew the drug was toxic to the heart, same mechanism of action to the liver, similar drug dose response. So basically, you have to show this. This is basically 15 years of use in patients to finally realize this, and in 2 weeks, we could tell you what the outcome was. So this is what the chip looks like when we print it. So this is our what we actually do is we now print the chips, and we print the chips with the organ tissue equivalents. And you can see here that these organ tissue equivalents are all printed. Is about the size of 3 times stacked together. This is liver, kidney, and ovary. And you can see the flow going through the slide. The only thing that is not printed here is the glass line and the plastic tubing. But this helps you to standardize the system. So we are now using this system. We had this $55 million program from DITRA and NASA, and NSF, National Science Foundation, to prevent the next pandemic, looking at different mechanisms of doing that. In this particular system, we're taking different viruses. We're infecting these organoids. We're looking at infection, replication, dissemination. We're looking at big data with crunch that data with AI so we can look at biomarkers and prophylaxis and treatments. These are some of the AI papers that we published in the last year and a half. These are 6 papers that we published using AI to show how this system is actually helping us to look at the differential expression, for example, of viral infections, or looking at different types of responses to these viruses. And we have another program with BARDA. This was about a $30 million program with BARDA, and we then just got it renewed for the second phase. We just completed the first phase, which was looking at chlorine, and the second phase is looking at other agents like mustard agents and other agents of the sort. This is the second. So this is from the chlorine program that we have, basically looking at how the chlorine, we can detect this down to 1 part per million. And by looking, so I'll show you how this works, but basically we are showing you the dosing on the far left. Then in the middle path, we can follow these organ tissue equivalents to look at what happens to the actual organoid. And we then look at long-term scarring. And from the long-term scarring, we can look at transcriptomics, metabolomics, and proteomics. And then and I always like to joke, we are an equal opportunity omics employer. We leave no omics behind. And then you can see with this all this data, we're able to crunch this data through AI, and then basically bring in, and we were able to assess over 100 medical countermeasures or drugs based on the targeting that we looked through this AI analysis. And then we were able to look at repurposed drugs, and we found 3 hits of drugs based on this that we're now testing to take to the next level. And so we now have the second BARDA program, which is another 5-year program, looking at other agents, as I mentioned. We have another program with NASA looking at radiation and regolith exposure. We have another program with CDRMRP looking at burn pit exposures. And we have another program with RARE and the VA looking at jet fuel exposures. So, and we have another program with BARDA, which is our latest program that was just funded this past year, looking at using this as an immune chip. So what we're doing here is we're integrating muscle, lymph nodes, lungs, and bone marrow together. So you can expose the lung, for example, to a viral agent, and then you can come in and do a vaccine to the muscle and basically compare what happens with these vaccines before and after you give these agents. All right, so now we're also using this for cancer therapy prediction. So as you know, current cancer therapy, one drug, many patients, right? And so the future here is one patient, one drug, using some of these strategies. Let me show you. Here, the concept is we have the patient, we take the tumor biopsy at the time the patient comes, we take a sample of that tissue, create an organoid with that tissue. We can then create a tumor in a chip, and we then can expose the tumor chip to these different drugs so we can know what the best therapy is. So we now have a bunch of trials at Wake Forest using this technology with support from the NIH and the NCI. Basically, what we have here is, for example, doing colon cancer, pancreatic, lung, brain, and other cancers. And that's exactly what we do. This is showing you, for example, for a GI tumor. And you can see here the differential treatment that is needed for these patients based on patient numbers. These are different patients, same type of tumor, but totally different response. And we now just had additional funding also from NASA to look at taking these into space. So the concept here, and this is now an active program where we basically take the tumor biopsy, create the organoids, do the chemotherapy, but send it into microgravity conditions so we can do the differential gene expression and look at what pathways are regulated. And why is NASA interested in this? People always say, why would NASA want us to send this into space? So in fact, we just had while this meeting was going on, we had a full day session with the FDA on this stuff and sending things to space. The reason for this is because these tumors, they get a much faster response up in space and microgravity. And therefore, because you get this early response, you can look at earlier expression genes. And so this is some of the work that we already published, looking at changing chemotherapy responses in microgravity. And you can see how you're shifting the treatment response to the left by making the tumors a lot more susceptible to these treatments. And by doing that, we can then allow these tumors to look at the pathways and see what's better. This is a simulated gravity for patients with colon cancer, and you can see here exactly what we're talking about in terms of shifting that curve to the left. So to summarize then, there are many ways to look at trials, right? We can do 2D cultures and low cost, rapid results, high throughput, but not very relevant often not relevant to clinical response. So they're a great model. I'm not knocking it, but in terms of clinical trials, we're talking about clinical trials. So small animals, reasonable, expensive, but again, limited physiological relevance. Large animals, expensive, slower, low throughput, sometimes challenging. Clinical trials, most expensive, the slowest there is, inconsistent sample data, low throughput. No system is perfect. Neither is this one. This system's not perfect. Still, no system's going to be perfect, but at least this system does give you a reasonable cost, rapid results, and high throughput and high clinical relevance. So I think it'll play a role. The FDA, just in May of this year, we had been working with the FDA for a little while in terms of looking at alternate systems. And finally, in May of 2025, they came out with this statement that you see here. And in October of this year, the FDA approved the very first drug to go into a phase 1 clinical trial with absolutely no animal data, based on chip data and AI. So these organ tissue equivalents can be used for drug discovery, toxicology, biosensing therapy applications. At our institute, we have a big portfolio of these tissue organoids that we can use for drug testing and personalized medicine and cancer therapy. And again, just to acknowledge our entire team at WFirm and to thank you again for having me. Appreciate it.
Thank you, Tony. Any questions or comments? Go ahead.